Okay, so first question. According to the fluid mosaic model in membrane, what can you say about the fluid mosaic model? Do you remember what the fluid mosaic model was? If all the above isn't in there? Who's number 3? Ooh. Answer is A. They float around in the lipid bilayer. Okay. Let's look at B and C. There is a double no, there's not a double protein layer. There's a double lipid layer, right?
See, that's where I got screwed up.
Mm-hmm. The proteins, remember, can either be all the way, like integral proteins went all the way through the layers, or you have the ones on the outside or inside. And then fats floating in the protein layer. It's not a protein layer. It should be there are proteins floating in the fat layer. That would work. Yeah, that's it. Okay, next one. Phospholipid molecules. What can you tell me about phospholipid molecules? This test? Probably not next week or the week after. Maybe at the end of the week, but Wednesday next week we have a short period, or short day.
Next week we have a short day?
Mm-hmm. Get out at noon on Wednesday
Why?
for in-service.
Why on Wednesday?
I
Is it
don't
Thursday, Friday?
know.
Why?
It's going to be like Hastings Public.
No, I want on Friday.
Answer is C. Let's look at A, B, and E. So the phosphate containing end is polar, and the fatty acid end is nonpolar. Think, okay, do fats mix well with water? No. Do you guys remember that water is polar? Can you remember that? Do you remember that? Water is polar. So if polar molecules are attracted to each other, so the fatty acids do not attract, so they have to be nonpolar. Because water is polar. All right. They are arranged in the double bilayer or double layer. Remember, it's the bilayer. And then, did anyone answer D? No, but E, we just went through. You took Brielle's favorite answer. Two functions of the cell membrane. I'm gonna, hold on. Ooh, this looks better. Answer is B. Yes. So produce mitochondria. It doesn't produce the mitochondria. The mitochondria is inside the cell membrane. So it is, yes, the phospholipid bilayer. B. That's the correct answer. Which of the following is not a component of the cell's membrane structure? So which of these are not found in the cell membrane? Answer is E. Yes. Glycogen. Glycogen is the molecule with glucose gets put into really long chain and then stored in like your liver for and then you can access it for energy. So that's what glycogen is. It is. So that is not in your glycerol. It is it's the backbone of the fatty acids. Remember lipids? It's a glycerol, and then usually had 3 fatty acid chains coming off of it. But for a phospholipid, which is part of the membrane, it's the phosphate group and 2 fatty acids. A glycoprotein that extends from the surface of any cell may do what? Think about what a glycoprotein was. Oops. I didn't mean to do that.
That's exactly what I thought.
So the answer is C. So remember, glycoprotein is a protein that has a carbohydrate chain coming off of it, and it would act as a receptor. And those receptors or markers, I guess it's a marker, not a receptor, and the markers would mark your body cell. You each have your individual the carbohydrate chain that's on your cells. Next, a cell membrane is composed mostly of, whoops, answer is E, yes. Lipids and proteins. Okay, that's it. So keep your clicker and we'll have questions at the end of this part. So we're going to look at movement through the plasma membrane, which that means that, well, we'll see. What? The cell membrane. Yeah. We'll see if we can do organelles and stuff. I don't know if we'll get to it. If we don't, then we're going to have to move the lab to Friday. We'll start the lab on Friday and move everything back. Yeah. Directly through, so you can move through the plasma membrane directly through the phospholipid membrane. There's membrane channels, which we talked about yesterday, carrier molecules we talked about yesterday, and vesicles. But we're going to look at specific pathways. So if you go directly through the plasma membrane, it can be through, it can be through diffusion, osmosis, filtration, mediated transport, which includes facilitated diffusion, active transport, and secondary active transport. We're going to go through those. Okay, so diffusion. Diffusion is the movement of solutes from an area of high concentration to area of low concentration. So remember, a solute is something that is dissolved within the solvent. So we're moving like salt across the membrane or moving an ion. And if it goes from high to low concentration, it goes down its concentration gradient, or it goes with its concentration gradient. And the concentration gradient is just a difference between 2 points. And then viscosity is how easily a liquid flows. So if the solvent is more viscous, it's going to diffuse faster. And if it's less viscous, it diffuses faster. More viscous would diffuse slower. No, syrup would be very viscous. It would have high viscosity. And so then diffusion would occur slowly. Low viscosity.
So it has high viscosity because
Yeah. So that example has like its sugar cube or salt cube down on the bottom of the beaker. The concentration of the salt would be very high near the cube and very low outside, and so just by random distribution of molecules, eventually it will equally spread itself throughout that beaker. Here would be another example where, okay, we talked about there being a barrier here, this creating a barrier. Because this is nonpolar, the middle is nonpolar, then it cannot, they're non-lipid soluble molecules, so it does not dissolve in lipids. It cannot go through that layer. But it could go maybe through a channel. It's going to have to go through a channel or some sort. Lipid soluble molecules are able to pass directly through because it's not repelled by this nonpolar area. And here it's showing the concentration gradient, so the high concentrations here, lower concentrations here, so it's going straight through the membrane channel. Remember that the outside of the membrane and inside are hydrophilic, and the inside of a channel would be hydrophilic, and the middle here would be hydrophobic. Osmosis is just like diffusion, except for it's the water molecule, which usually is the solvent. So you almost have to think of it backwards. If you have a high concentration of salt, that means you have a low concentration of water. Low concentration of salt would mean a high concentration of water. So it's almost, you think of it opposite. And water moves from an area of low concentration of solute to an area of high concentration of solute, because when there's a low concentration of solute, that means there's high concentration of water. So it's still moving down its concentration gradient, just in comparison to the water concentration gradient, not the solute concentration gradient. Osmotic pressure is a force required to prevent water from moving across the membrane by osmosis. So if once the pressure on one side of the membrane gets high enough, it will stop the osmosis from occurring. There's some comparative terms used to describe the osmotic pressures. So we have isoosmotic, hyperosmotic, and hypoosmotic. Isoosmotic is solutions with the same concentration of solute particles, so it's already in equilibrium. The concentration of water would be the same also. Solutions with greater concentration of solute is hyperosmotic. And solutions with lesser concentration of solute is hypoosmotic. So in a hypoosmotic solution, if you put, well, do we have examples here? Well, look, okay, so in here, this would be, has low concentration of solute, so that would be hyperosmotic, right? So what's going to happen since there's a higher concentration of water outside, this is the membrane here, and low concentration of water inside, the water is going to flow into that tube and cause the level to rise until the pressure of this liquid pushing down on the membrane reaches the osmotic pressure where the water can't go in anymore. Okay, this is important in cells because your blood is has a concentration of solutes, which is why, like, if you go to the hospital and you need fluids, they don't just give you straight water into your blood vessels when they put you on an IV, do they? It's always saline solution, and that saline solution has to be the right concentration so that...
The close-up meeting is beginning in the auditorium.
Does anyone need to go? I think so. What? I don't... You can leave it. Yeah, I don't know. So you always have to make sure you have the right saline solution, otherwise it will make an imbalance of water concentrations and can affect your blood cells. So you need an isotonic solution where the water doesn't go into or out of the cell and it would neither shrink nor swell. A hypertonic solution, so let's look at this hypertonic solution. What did hypertonic mean? Or I guess hypertonic means that you have a higher concentration of water inside. So here's a hypertonic solution. There's a higher concentration of the water inside the cell than outside the cell, so you must have had more salts or more solutes outside the cell. And so the water leaves the cell trying to equal out the concentration. And when it does that, it shrinks, and that is called crenation. That whole process is called crenation. Crenation with an N. A hypotonic means there's low solutes in the outside solution or a high concentration of water, and so the water would rush into the cell and it would cause the cell to expand, and the cell would eventually expand until it would burst, and that process is called lysis. So you need to know those different solutions. Okay, filtration works like a sieve, just like when you guys filtered the carbohydrates that you filtered? Proteins, right? In the protein lab, you set up a filter. Just like that. It depends on a pressure difference on either side. So your pressure outside was pushing your solution down through the filter paper. And it moves from the side of greater pressure to lower pressure. This is very important in the kidneys, and that's how when the blood goes past, it builds up pressure and pushes the waste through to the kidneys, through all those tubules. So water and small molecules are able to move through the membrane while large molecules remain in the blood. Mediated transport would involve the carrier protein. We talked about carrier proteins yesterday, or channels in the cell membrane, and they are specific to a type of a molecule. So it shows that on that top picture where there's a specific binding site that would fit a specific molecule. This molecule would fit, but this molecule doesn't. There is competition. There may be two different molecules that could fit in the same. So it would be, they would be competing with each other for space to go through the carrier protein. And then saturation, the rate of transport is limited to the number of available carrier proteins. So if you only have a few molecules going through, the carrier protein can keep up with the workload. But as you build up that concentration, it can only go so fast. Once you get to a certain rate, it can't go any faster, and the buildup it'll build up outside the cell, which is what this is showing. So when it's low, it can easily transport these molecules. Even it gets to a certain point where it's able to transport it at a fast rate. Once there's more solute, though, or the molecules that need to be transported, it can't go any faster. So if you were to graph that rate, it would increase until it reaches a certain rate, and it can't go any faster. It plateaus. That's what this is showing. We'll go through that. Okay, facilitated diffusion is a passive form of mediated transport, meaning it doesn't require any energy. You're able to move large water-soluble molecules or electrically charged molecules across the plasma membrane, such as amino acids and glucose in, and then you can manufacture proteins can go out. Right, passive means it does not require energy. Whereas we have active transport would require energy. So facilitated diffusion, it's diffusion. It's still diffusion. It's just being done through a carrier protein. So it is still going from high concentration to low concentration. Whereas active transport, you are going against the concentration gradient, and it requires ATP, requires energy, in order to push molecules out into or out of the cell where the concentration is already high. So the rate of transport not only depends on the concentration of the substrate that it's moving, but also depends on the concentration of ATP. And we kind of already talked about this yesterday, right? We talked about the active carrier molecule. And this is an example, the sodium-potassium pump, which is a very important channel. It creates electrical potentials across the membrane. So we'll go through this step by step. So you have sodium inside the cell. You would have 3 sodium molecules be able to bind to this carrier protein, and ATP binds here. Once ATP breaks and releases its energy, it changes shape, releases the sodium outside the cell, and then there's active sites that are exposed for potassium to bind. And when the phosphate group leaves, then the potassium binds and it changes shape and releases out into the cell. So it has 3 sodiums going out, 2 potassiums coming in. So is that symport or antiport? Antiport, because they're moving in the opposite directions. Remember symport, we talked about symbiotic relationships. They work together, so they move together, whereas antiport, they move opposite. Secondary active transport, you would have 2 proteins that would work together. So something that one protein is transporting, another protein would use to transport a different molecule. We already talked about movement of glucose. So here, glucose, so the sodium-potassium pump pumps out the sodium. The sodium, some of the sodium can be used in this this protein. It binds the sodium and the glucose. It needs both in order to change shape and release the glucose in the cell. So would that be symport or antiport? That would be symport because they're both going into the cell, same direction. So yeah, the sodium-potassium exchange pump maintains concentration sodium inside the cell, so it takes active transport to get the sodium out here because there's more sodium outside the cell already. But since the concentration gradient here is high for sodium and low for sodium in here, it's a passive transport going through here. So it does not require ATP in this case. The concentration gradient for sodium provides the energy required to move glucose against its concentration gradient. So you don't require ATP. Just showing that a little bigger. Endocytosis. You're bringing substances into the cell, and you bring it into the cell using a vesicle. And so there's phagocytosis, which is shown where the cell kind of puts out these little projections of the cell membrane and engulfs a foreign substances and brings it in. Pinocytosis is a specific type of endocytosis where you're bringing in liquids. So pinocytosis brings in liquids. And then there's also receptor-mediated endocytosis. So pinocytosis, we already said right here where it's going to bring in liquids. Actually, this looks like a picture of exocytosis, doesn't it?
No, it's on that
Oh.
side.
Well, this is exocytosis, it looks like. But receptor-mediated endocytosis means there's receptors on the cell membrane that would actually bind the molecules that it's bringing in. And once you have these molecules bound, it creates a little vesicle and pinches off some membrane, and it comes inside the cell.
So what is receptor?
That's what it is.
That's what that is.
Yep, that's receptor-mediated endocytosis. Exocytosis is the opposite of endocytosis, so it's expelling something outside the cell. But you still have a vesicle. It would come up to the cell membrane, join, and then just open up. And some examples: secretion of digestive enzyme by the pancreas, secretion of mucus by the salivary glands, and secretion of milk by the mammary glands are all through exocytosis. Okay, grab your clicker. Well, you have your clicker. So number one, or the first one, which of these molecules is correctly matched with the method of movement? When it says dissolved in the double phospholipid layer, that means it's able to pass straight through without using a channel. Oh, you all have answered. That's right. You have one less. Answer is C. C. So large water molecules, water-soluble molecules are transported by carrier-mediated processes. So lipid-soluble molecules, lipid-soluble means they're able to go through that nonpolar region in the membrane, so they're able to go straight through the membrane instead of using a channel protein. The water-soluble molecules in B would mean they cannot go through that nonpolar layer, so they have to use the channel protein. So those two are basically swapped. And glucose and amino acids, they pass through not membrane channels. They have to be their carrier-mediated processes. So membrane channels are those ones that are just open, and you go with your concentration gradient. But carrier-mediated means that the protein is the one that changes shape and attaches, and then it would release it. Okay. Let's try the next one. Can you tell me about diffusion? got it. Answer is D. D. So A and B. Let's look at.
I think the confusion was from low areas of concentration or higher
High to low.
high to low.
Yes. So A would be correct because it's area of high concentration to low concentration.
You spray something in the room.
Disperses, yep. Because there's high concentration where you sprayed it.
And low everywhere.
Yes. And when you spray something in the room, it's because of random movement of the molecules that it's able to spread. So that's why B is correct. And it always produces net movement. It would be with the concentration gradient, not against the concentration gradient. So it's D, A, and B. Which of the following statements concerning solutes and solvents are true? Okay, the answer is A, yes. So it's a solvent would be the one in greater concentration, and it dissolves the solute. So it is the predominant liquid or gas. D. No, all solute particles move into or out of the cell by diffusion. Not if they're non-lipid soluble. If they can't pass through that non-polar layer, they can't. And then also, sometimes the carrier molecules there aren't the right carrier molecules for those for the solute particles. So sometimes they're moved into or out of the cell by active transport or mediated transport. Red blood cell placed into distilled water. So distilled water has no solutes in it. What would happen?
Nothing in it.
Just pure water, no solutes. Okay, the answer is A, yes. So since there's higher concentration of water outside the cell, then there's more water outside the cell, it's going to rush into the cell, which make it swell. And when it swells and breaks open, that's called lysis.
What's
Crenate would be when it shrinks. Crenation.
Then wouldn't it be using what's inside the cell to make it even?
No, it's gonna. For red blood cells, you're talking about osmosis, so the water concentration.
So when would a cell
Right. If you put it into very, very salty water, where the concentration of salt is higher, so there's lower concentration of water, the water inside the cell is going to come out of the cell, and it would shrink. What's true about active transport? answer is E. Yes, all the above. It would be all of these. And that's it. So I think tomorrow we don't have time to get through organelles. So tomorrow we'll talk about organelles, cell metabolism, protein synthesis. Then we'll do the structure of cells lab starting Friday after your quarter quiz. And we will have a quiz over the notes up to this point, probably Tuesday. Next